This video tutorial looks at pyrosequencing. In the first decade of the 21st century, a variety of approaches were used to develop fast, cheap methods to sequence genomes. One of them is pyrosequencing. This method was developed in 1996 and uses sequencing by synthesis, not by chain termination, as in the Sanger method. It involves synthesizing a single strand of DNA complementary to the strand to be sequenced, one base at a time, whilst detecting, by light emission, which base was added at each step. So first of all, a long length of DNA to be sequenced is mechanically cut into fragments of around 300 to 800 base pairs using a nebulizer. These lengths are then degraded into single-stranded DNA, shown as ssDNA. These are the template DNAs, and they are immobilized. A sequencing primer is added, and the DNA is then incubated with a number of important enzymes. Only one of the four possible activated nucleotides, so that's ATP, TTP, CTP, or GTP, is added at any one time, and any light generated is detected. So one activated nucleotide, so that's a nucleotide with two extra phosphyl groups, such as TTP, thymine triphosphate, is incorporated into a complementary strand of DNA using the strands to be sequenced as a template. As this happens, the two extra phosphoryl's are released as pyrophosphate, so that's PP. In the presence of APS enzyme, that's adenosine phosphosulfate enzyme, the enzyme ATP sulfurylase converts the pyrophosphate to ATP. In the presence of this ATP, the enzyme luciferase converts luciferin to oxyluciferin. This conversion generates visible light, which can be detected by a camera. The amount of light generated is proportional to the amount of ATP available, and therefore indicates how many of the same type of activated nucleotide were incorporated adjacently into the complementary DNA strand. Unincorporated activated nucleotides are degraded by another enzyme, and the reaction starts again with another nucleotide. 1 million reads occur simultaneously, so a 10-hour run generates 400 million bases of sequencing information. Software packages assemble these sequences into longer sequences. I'm also just going to quickly mention bioinformatics. Now, a branch of biology called bioinformatics has grown out of this research to store the huge amounts of data generated. It would have been impossible to store and analyze these data prior to computers and microchips. Software packages are specially designed for this purpose.